Dr. Watkins had spoken at one of our invitation meetings. Her resume, as you can see, is 110 pages. She's on the medical advisory board of the Migraine Research Foundation. She's associate editor of Brain, Behavior, and Immunity. She's involved with a pharmaceutical company, bringing a Glia modulator to market. So this is really an honor to introduce her today. Thank you.
Thank you. It's a pleasure to be here. So what I want to do today is tell you about a story that we're really excited about. It's one that we've been working on for the last 25 years. And what the story is going to show you is that non-neuronal cells called glia are critically involved not only in the creation and maintenance of pathological pain states, but also in dysregulating the actions of opiates so that they can't do the job we want them to do for pain control. So what I want to do is interest you in just a few global concepts. The first has to do with how we conceptualize pathological pain. In pathological pain, hot, cold, hard pressure pains are greatly amplified. Warm, cool, and light touch are now perceived as pain. Classically, every model used to try to understand such pain is focused exclusively on neurons. Immune-like glial cells, immune cells were not thought of because they weren't thought of in terms of communicating with neurons. But this view has dramatically changed because work over the last 25 years has clearly documented that these non-neuronal cells are key players, both in the creation and maintenance of pathological pain states. But in addition to that, work over the past 10 years or so has also demonstrated that they become activated in response to opiates and prevent opiates from having the desired pain-relieving effects. Now, the data I'm going to show you today I think are really profound because what the data say is that if we target these non-neuronal cells called glia, if we can target these immune cells, we should be able to far better control pathological pain states as well as improve the clinical efficacy of drugs like opiates for pain control. And what personally I think is very exciting, I'm going to share some of this with you today, is that there are now, for the first time, glial targeting drugs approaching clinical trials. So this exclusive focus on neurons up until recently was really very reasonable because the concept has always been that if you have peripheral nerve injury, you have amplification all along the pain pathway, and it's always been thought that the neurons are to blame. But over the course of the last 25 years or so, a new player has emerged that within the trigeminal system, within the spinal cord as shown here, there's another player. And that these are these non-neuronal cells called glia, and they get all hot and bothered under conditions of pathological pain, releasing substances that cause pain amplification. So, if this is true, what do the data say? Across the world, across laboratories, across models, what the data say is that these non-neuronal cells become activated in spinal cord and trigeminal pathways and pain pathways under conditions created in every single clinically relevant animal model of pain. Everyone. But far more important than that is that if you block the activation of these non-neuronal cells called glia, if you block their actions by blocking the actions of what they release, you will prevent pain in every single clinically relevant animal model. And it doesn't make one oblivious to pain. Pain doesn't disappear, it returns to normal. What this does is it removes the amplification of pain beyond what is supposed to be present. And beyond pain, what the data say across labs, now across the world, is that if you block glial activation, you will make opiates work better for pain control. Opiates work better, stronger, longer to suppress pain. You prevent tolerance, you prevent dependence, you prevent reward that's linked to drug abuse, and you will suppress other types of opiate-induced side effects, so problematic to chronic pain patients on opiate drugs. But this has got to sound really, really strange, because if you've ever thought about glia, which virtually nobody ever has, if you ever thought about these cells, this is the picture of glia that you have under the microscope. Little bodies, little arms and legs, look like little statues under the microscope, just sitting there. But now that people can image living, healthy, intact brains, that's not the image of these cells at all. This is. These cells are constantly moving. If you take an amplification, magnification, one of those little arms, maybe it's a little leg, who knows, they're stretching out, they're contracting, they're feeling up the entire extracellular fluid, all the neurons, every 2 minutes throughout your brain, throughout your spinal cord. That tickles once you start to think about it. But if you now think about these cells, and they're actively moving, looking for problems. And on the right screen, at the tip of the pointer, this is only going to happen once, so I need you to watch this point. This is what happens when these cells find the danger, the damage, the problem that they're looking for. They attack. Isn't that cool? No. So let's return to the slide for a moment. What I said was that they become activated under every single clinically relevant animal model, and that's true, you can see it. But what I also said was that if you block their activation, you will prevent pain in every single clinically relevant animal model, but there's a problem. And the problem is the drugs that have been used up to this point. They all have off-target effects. In fact, they also affect neurons. So maybe all of this is bogus. Maybe these glial cells have nothing to do with pain whatsoever. It just has to do with these drugs having off-target effects nobody suspected. So how can we get to the root of the question? Dreads. I love this stuff. Designer receptor exclusively activated by designer drugs. Translated, fake receptor, fake drug. That fake receptor I can express in any cell type I want, including just glial cells. And that fake drug will bind only that one fake receptor. So I can turn on, I can turn off glial cells for the very first time exquisitely, exquisitely precisely. So what is it now going to say? So what is it going to now reveal when we only turn on or only turn off this cell population? So the concept is, if we take microglial cells and we express these receptors only on the microglial cells, we now give them this fake drug that only sees that one receptor and it binds it. The concept is, if it's an activating receptor, that should now create an activated state. The cells should theoretically release all this stuff that should amplify pain, like interleukin-1. This is one of the things that they release. So what's the data? On the y-axis is pain. If you go down on this axis, pain is increasing. This is going across time. Control animals, given this drug, it does nothing across time. But if I now give them an excitatory receptor only on microglial cells and activate only those cells, you see a fall in pain threshold. Touch becomes pain, and this animal now experiences pain when only microglial cells are turned on in the spinal cord. Now, the much more important question, actually, is that what is it that they're really creating this pain with? And indeed, if you block interleukin-1, that's one of those pain-enhancing things the glial cells release. If you block that in spinal cord, no such pain enhancement occurs. Now, the far more important question actually is what happens when the glial cells are already all hot and bothered, say under conditions of chronic pain. If we only take them out, if we only inhibit those cells, what will happen to pain? So in these animals, we know that we have a condition where we have amplification of pain. These glial cells are activated. So the concept is, if we now activate inhibitory receptors, that should make those glial cells calm down and theoretically should quit putting out all this pain-enhancing stuff. So what do the rats say? Well, you have these rats, you measure their pain threshold, and now after nerve injury, you see a fall in pain threshold over time. You give them a control group, this drug, it does nothing. But if they have an inhibitory dread, that receptor I can activate only on glial cells, and now I turn those cells, only those cells off, pain goes away. This is the first demonstration that in reality, for real, these cells are really critically important in terms of amplification of pain. So what is it that they start releasing when they get activated? Well, they start releasing a whole host of things that people who study pain immediately recognize as pain-enhancing substances. But in addition, what they do is they amplify pain messages from your body to your spinal cord. So if you step on a nail, you now release way too much pain transmitter into the spinal cord. And in addition, your spinal cord gets all jazzed and amplifies that message on up to the brain. So you have double amplification. Amplification in, amplification up, and the world is not a nice place. Of these, the pro-inflammatory cytokines is a family of excuse me, very pesky proteins that are amplifying pain and are one of the major products that these cells are releasing. So, what causes the problem? All of these things can activate glial cells, but given our short time together, I want to just focus in on two of them, on endogenous danger signals and opiates. What opiates are, like morphine, oxycodone, and so on, is very clear. What are endogenous opiates? Well, endogenous opiates are little bits of you. Little bits of you. That's not supposed to be floating around free in the extracellular space. Little bits of your cell membranes, your DNA, your proteins, bits of your blood that's not supposed to be in the spinal cord floating around free. Things that say you have damage, you have death, you have danger. That's what endogenous danger signals are. And in terms of what they're involved in, they're now implicated in a wide range of pathological pain states. Basically, you can think of this as when bad things happen, endogenous danger signals, things that say danger, stress, damage, death, endogenous danger signals get produced. They activate glial cells. Glial cells release pain-enhancing substances, and away you go. Opiates. Well, it turns out glial cells don't like morphine. Glial cells don't like methadone either. So if you have a rat and you're testing a radiant heat on the tail or on the paws and so on, if it's just a control group, very boring animals, this is time, across time, they respond. What basically is that if you have a radiant heat on a rat paw, normal conditions they'll flick, they'll remove their tail or paw in about 2 seconds. But if you give them a really good analgesic like morphine or methadone, it suppresses pain. So it now takes, maybe 10 or 12 seconds before the animal recognizes there's a painful stimulus and moves its tail or foot. So this increase in latency means they're not sensitive, they're analgesic. They've lost the ability to sense pain. And for both morphine and methadone, very good analgesics. You see this analgesic response that then fades away across time and appears to be gone at about 100 minutes or so. But notice I said appears to be gone, because it's not. You just can't see it. Because if you give something that will block the actions of glial cells, like blocking interleukin-1, that pesky protein that is amplifying pain, and you block it now at the level of spinal cord, analgesia comes right back. It's not gone. What we think is going on is that when you give things like morphine or methadone, you produce pain suppression. They're analgesics. And that pain suppression should go away as the morphine's degraded and cleared. But what's perverse is that when you give opiates, you also activate glial cells that release pain-enhancing substances. So what you feel, what I measure in the rats, is the summation of pain suppression by morphine and pain enhancement by the glial cells. They're fighting in opposition. So when you think this additivity between pain suppression and pain enhancement, when you think the analgesia is gone, it's not. Because if you block the glial cell, the analgesia comes right back. Morphine can't work as well as it should because it's activating these glial cells as well. But what is really exciting is that the receptors involved, how the cells respond to opiates on neurons and on glial cells, are different. There are different receptors involved. And this has to do with opiates. A lot of opiates exist as, back to the last lecture, mirror images of each other. So they exist as left hands and right-handed molecules. They're not the same. They're backwards from each other. Things like morphine, and plus is a very good analgesic when it's in this minus or left-handed isomer, and it has no effect if it's the opposite hand. It's like sticking your car key in the wrong ignition. It might fit, but it won't turn it on because it's the wrong fit. It all has to do with fit. So the minus morphine, minus oxycodone, and so on, is powerful analgesic, and the plus is little kitty has no effect. Okay? This all has to do with fit. So if you have minuses and pluses, what the receptor on neurons is very stereoselective, meaning it only binds minus. That's the only thing it can bind. It simply doesn't fit. Same thing for the antagonists. Narcan is naloxone. naltrexone. Those are antagonists at the receptor, but only if they're in the minus configuration. If it's in the plus, it doesn't fit. And this is critical for everything we're going to be talking about in just a second. And why it's critical is glial cells are different. Glial cells are equal opportunity employers. They don't care if it's plus or minus. They both fit just fine. They both activate glial cells. They both create pro-inflammatory cytokines. They both cause problems. But fortunately, this also holds for the antagonist. And why that's important is that for the first time, the glial cell receptor can get blocked by, can get constipated by the plus version. And why that's important is why that says is we should make opiates work better for pain control by either this should work better for pain control because it doesn't block morphine from pain suppression because that's through the neuronal receptor. It doesn't fit. But it does block the glial activation that opposes the opiate analgesia. And this is true. So if we give morphine, for example, to rats, now you see this increase in pain threshold. That means they're analgesic. They're not responding to pain very well. And it then fades away around 100 minutes or so. If we add this plus naloxone or plus naltrexone that does not bind the neuron receptor, you make it better because you take away the glial cell component that's constraining the ability of opiates to cure pain. And why that's important is as soon as you say you have different receptors, that means we can target it. We can target it by developing new drugs, better ways to separate the actions of neurons from the actions of opiates. So to make opiates work better, you will either want to modify the opiate so it doesn't see that pesky little glial receptor, or you're going to create a longer lasting version of plus naloxone, which is one of the things my small company is working on, my lab has been working on for 15 years. So that is one of the directions I'll be telling you about. But for us to target it, we got to know what this receptor is, and it turns out, after a very long story, that it turns out to be this ancient immune receptor called toll-like receptor 4, TLR4 for short. You can think of this as a not me, not right, not okay receptor. So this receptor sees things like endotoxin, like bacteria. It also sees things like endogenous danger signals, little bits of you that say you have cellular stress, damage, death, things that are warning signals you have a problem. And we also discovered that it sees every single clinically relevant class of opiates. And we've done a lot of work on this. But it all fits the following model. That is, if you give it a drug like morphine, it works at two places, not one. It works on neurons in order to amplify, to produce analgesia, but it also activates glial cells to produce things like interleukin-1 that suppress the ability of opiates to control pain. But if you give something like plus naloxone or plus naltrexone, it plugs up the glial cell receptor, can't touch the neuronal receptor, and now when you give things like opiates, they work unopposed to produce beautiful analgesia because the glial cells can't compromise it anymore. So we've just said that this toll-like receptor 4 also sees endogenous danger signals, little bits of you. But if that's true, what that would suggest is that this approach may also be a standalone treatment for pathological pain because that's getting created by things like endogenous danger signals. And that's true. So in rats, before surgery, they escape from calibrated pressure on the paw at a certain force. Now after surgery, you see this fall in pain threshold, touches become pain. If you give them a vehicle of the spinal cord, it does nothing. But if you block toll-like receptor 4 in the spinal cord, neuropathic pain fades away and it's gone. And it's not just pain. If you're on opiates, you worry about drug abuse. Drug abuse kills. Every single time people have created drugs that make them work better for pain control, because there have always in the past been working with the same receptor that's important in drug abuse, you also amplify drug abuse. This is a problem. So we were worried about that. So we wanted to see whether or not we made a super opiate, we also made a super addictive one. Fortunately, not. I'll show you that that's actually not true at all. But let me introduce this. We've done this a variety of ways, both behaviorally as well as neurochemically. I'll just tell you a little bit about the behavioral version. This is called conditioned place preference. This is kind of the concept that if you go someplace you've never been before and something evil, awful, terrible happens to you there, you don't like that place and you avoid it. But if you go someplace you've never been before and something just exquisitely wonderful happens to you there, you like that place. It feels good to be there. Rats are the same. You bring in rats. You let them explore 2 different contexts they've never been before, and then in one of them, they get a good drug like morphine or saline, and in the other one, they get just saline. You bring them back another day and say, okay, rat, where do you like to be? And if they had saline on both sides, they still don't care, one side or the other, but if they had a really good drug on one of the sides, you basically can't peel them out of there. It feels good to be there. So we now you're measuring the increasing amount of time they spend, they choose to spend in a context where they got a really good drug, and you see an increasing amount of time the rats, the guys, spend in the context that they got a really good drug like morphine, but if you block toll-like receptor 4, no such increase occurs. And we've done this a lot of different ways with a lot of terrific collaborators. We know that if you block toll-like receptor 4, you can block neurochemical indices of reward, condition place preference, self-administration, incubation of craving, reinstatement, relapse. That means falling back to taking drugs. It will also block that. And it's not just opiates, it's also cocaine, it's methamphetamine, it's alcohol. A lot of drugs of abuse can be blocked. And it's not even just drug abuse, it's also brain death. This is a new area for us. It's just really exciting to me. So you're stuck, you're captive. So you're going to get like 2 seconds on this. This is a heart attack cardiopulmonary resuscitation model in rodents. And if you have a heart attack and somebody jumps in and gives you cardiopulmonary resuscitation, your brain just really doesn't like that. And in areas that are very sensitive to loss of oxygen, parts of the hippocampus, for example, 45% or so of those neurons die. But if you jump in like an ER doc 30 minutes after a heart attack cardiopulmonary resuscitation and you block toll-like receptor 4, endogenous danger signals, stress, damage, death, you will prevent half the neurons from dying. Very exciting. I'm really excited about that. Isn't that cool? I thought that was really cool. So, summarizing what we've been through so far, what we said is that what the data say is that if you suppress glial activation, you will suppress pathological pain in every clinically relevant animal model, make opiates work better, block tolerance, block dependence, block reward, block side effects, da-da-da-da-da-da. All good things. So, summarizing visually, what we've been saying is that you have peripheral inflammation, peripheral nerve damage, you have amplification all along pathway. These hot and bothered glial cells are putting out all this stuff that amplifies pain, and what's perverse is if you give something like morphine, the little red pill, you make things worse. And you make things worse because the opiates are also activating the glial cells. They put out more of this pain-amplifying stuff, and pain becomes worse. My lab wants the little blue pill. That's what my lab wants. Now, my lab is after the little blue pill that's going to suppress the activation of these hot and bothered glial cells to be a standalone treatment for pain, but also to allow opiates to work better for pain control. And whether that is what we're working on with plus naltrexone to stuff up that particular receptor on glial cells, or an interleukin-10 gene therapy that's about to go to the FDA that I'll be telling you about later in the talk, this has been what we've been working on for the last 15 to 20 years. So I want to take a sidestep to a new issue that I think is really relevant to this group, and that is that we've been talking about these glial cells up to now as if they've just been in 2 states, this basal state, this boring state, or this activated state, releasing all this pain-enhancing stuff. But in fact, there's a 3rd state, and the 3rd state is called primed. And the prime state is kind of like I walk up to you and then suddenly, without any notice whatsoever, I slap you across the face. I get away with that the first time. You're not going to let me get away with it the second. Glial cells are just the same way. So if you get them activated the first time, they can enter this prime state where they're ready. They're waiting. They're ready. And if you activate them the second time, they explode into actions faster, stronger, longer than before, putting out way more of this pain-enhancing stuff. And so what we're wondering is whether this could set the stage for acute pain becoming chronic. And from our data, we think this is a major contributor. And from the literature at large, there's a variety of ways you can enter the state. Aging, I hate to say this, but from about middle age on, your glial cells are becoming primed. All those taxes, politicians, the neighbor's dog barking at 2 a.m. All these things are making your glial cells primed with stress. And aging and stress, see above, trauma, and opiates can all prime glial cells. So this is a 2-hit model that we're working with. The first hit are things that can create priming, aging, stress, trauma, inflammation, opiates. And then a second hit, appropriately timed, to say, well, what does that do to pain? And so what we're doing now is, we've done a lot of work on this. I'm just going to give you a couple little snapshots of the kind of work, is that we know that if we give these kinds of first hits with second hits, we can change no pain to pain. We can change pain to chronic pain. Okay, as an example, with the first hit, this is, as an example, this is laparotomy. This is done thousands of times in the United States every year. Exploratory abdominal surgery. In the rats, it was a model developed by a surgeon. You open up the rat, you run the guts, you peer inside the body, you stuff everything back in, and you go home. Well, he goes home. Sew him up, of course. And then weeks later, we now give them a second challenge. And what happens? Now, these are just boring control groups, just saying their pain doesn't change across hours. You have an animal that had a sham surgery, no real surgery. You give them this very, very mild second challenge, nothing happens. But if they'd had surgery, laparotomy, weeks before, you give them this identical challenge, now you see fall in pain threshold, touches become pain. And now it starts to resolve over time. So no pain has become pain. So what happens if you had pain? Can you now create chronic pain? So same first hit, but now you increase the second hit. It's a sterile bladder inflammation, not infective. You just irritate the bladder lining. And when you do this in an animal that did not have surgery before, you see this mild fall in pain threshold that now resolves over time. But if they'd had laparotomy surgery, a challenge, weeks before, and now you give them the same challenge, far deeper fall in pain threshold, 3 months later, there's no resolution of pain. Acute pain has become chronic. And what's really exciting is that if we block glial activation at the time of the first surgery and now give them the second challenge, it's as if they never had surgery at all. We can prevent this acute pain from becoming chronic. So we're studying what causes it. Can we reverse it? I'll show you in a minute. We now know how to now stop this thing in its tracks and go back to normal. But if you pause here for a second, what you're going to think is, oh, you know, there's something really deviously problematic with what she's saying. Very disturbing. And what the thing that's disturbing is when do you get opiates? After trauma. Trauma activates glial cells. Opiates activate glial cells. Isn't that a first hit, second hit hypothesis? Doesn't that suggest that there may be some really unpleasant side of opiates we've never really examined? Turns out to be true. So now we have the first hit being trauma. The second hit is now a short course of morphine after trauma, which is when you would get morphine under normal circumstances in response to trauma. Come on, you can go into the next one. There we go. Okay, so that came up a little too fast. So here in the squares are sham-operated animals. This bar tells you the time, 5 days of morphine after trauma. The animals that were sham, you remove the opiates, yeah, you get a little bit of withdrawal, decrease in pain, a little bit threshold, have a little bit of sensitivity, very mild, goes away very quickly. Animals that had nerve injury, you see a fall in pain threshold that resolves along this time course. But if you now have morphine for 5 days right after trauma and now track out the pain across time, massive amounts of increased duration before the neuropathic pain goes away. And we know it's not due to opiate receptors. We can take away the opiate receptors. We can do this with just activating toll-like receptor 4. We can recapitulate all of this. We know this is not due to opiate receptors. It's due to toll-like receptor 4 in microglial cells. So we know that if you have these 5 days of morphine, you see this amplification of pain. But now, if we just block microglia with that dread that I was telling you about, that designer receptor exclusively activated by designer drugs, it's as if they never had morphine. But keep in mind that if you take out the microglial cells on acute morphine, it works better for pain control. It works better for pain control and then prevents the long-term deleterious effects of opiates. We're very excited about this direction. It's the same result when you block toll-like receptor 4. But what perhaps is even more critical, because a lot of times you're not in control if you get opiates right after trauma, is can we reverse this? And the answer is yes. If we block either just microglial cells, or if we block toll-like receptor 4 for about a week, we can break the cycle of pain and then goes back to being normal. So, 25 years in the making, this is finally approaching human clinical trials. The Salute Therapeutics, as was the acknowledgement on the very first slide, was founded in 2009. I'll tell you about that in a second. In order to take research from my University of Colorado Boulder lab to take it to human clinical trials. We've been talking about plus naltrexone thanks to the Department of Defense deciding that that was in the national interest. Thank you, Department of Defense. They've awarded us about $5 million to take it to investigational new drug status for human neuropathic pain and drug abuse. What I'm going to tell you about now is something we've been working on for 15 years, and it's interleukin-10 anti-inflammatory cytokine that's just about to have the submission to the FDA to start clinical trials for neuropathic pain and also osteoarthritis. I'm going to show you some dog videos, which are very close to my heart about how well it's working. And thanks to the American Kennel, oops, that should be Kennel, not Kenner Club. One of my collaborators is named Kenner. I guess that's how it slipped in there. I'll show you that in a minute. So this is all focused on the fact that I've been telling you about these glial cells get all hot and bothered, releasing pro-inflammatory, pain-amplifying stuff. So with all the body of evidence, it looked very reasonable to try to attack it with your own anti-inflammatory cytokine protein. You make this. It is a protein called interleukin-10. Interleukin-10 is anti-inflammatory. Nature's own negative feedback on all the bad stuff that happens. You just don't make enough of it when you're in chronic pain. And it's also there's a lot of other symptomologies like arthritis, which have the same kind of pro-inflammatory milieu, so we were also testing it for things like that. So in 1992, we started doing this research. 2000, we started working with interleukin-10. 2009, because all this work looked so intriguing, Salute Therapeutics was formed. Salute as in to your health, X is for interleukin-10. Looks like Zudlug, but it's actually Salute. 2015, it's going to make it to investigational new drug status, which is so many, many years in the making. So let me tell you just a little bit about this approach, because this started, again, as a basic research study, only because I couldn't afford to buy this protein for my rat study, so I had to get the rats to make it themselves. And that's really how it all started. It just looked so cool, we had to take it to people. So we gave the rats an injection, lumbosacral space, like a lumbar tap, but in reverse. Put a little bit of stuff right in the CSF. No cut down, no surgery, just an injection. And why we chose interleukin-10 was it takes that entire family of things that are causing pain to go bad, and it suppresses all of them at multiple levels of regulation. So let me show you why we're excited about this from 1992 on up. So the guys on top are just boring control groups. This is 3 months, just showing you that testing over 3 months, they're very stable. The animals in red are the ones that are really exciting. They have this fall in pain threshold after nerve injury. Touch has become pain. Now we give them one injection, repeat, one injection, and one injection of this non-virus, who did away with all the viruses that cause all these problems, and neuropathic pain goes away, and it is gone for 3 months with one injection. And it's across multiple pain models, And it's not just rats, it's dogs. Any dog people here?
Service dogs.
Service dogs, I love service dogs. Dog people, this works in pet dogs. These are pet dogs who would have been killed by their owners because of chronic pain. Nothing works. Nothing works in people. Nothing works in dogs. Dogs get the same chronic pain just like people do. We now have dogs, and more dogs. This is the wonderful man I'm in love with as a collaborator, not any other level. This is just make sure my husband, if he hears about this, doesn't misinterpret. This is Rob Landry. He's an amazing chronic pain specialist veterinarian in Lafayette, Colorado. And Rob heard me give a talk and said, we have to get together. And so because of Rob's leadership and Rob's clients and everything that Rob does marvelously, we've done open label and closed label blinded studies of dogs. I'm going to just show you a couple of, just some bits and pieces of our data to show you what we're trying to do here. But what's exciting is this is real disease. This is not a rat who has been born and bred to be perfect. And it's not a rat who had something done to it and 2 weeks later you're trying to pretend this is chronic pain. These dogs have been in pain in years, and they've been on all the drugs, and all the drugs fail. So they are the proof of the pudding. So just examples of the kinds of things we're doing. This is Katie. You'll see a video of Katie in a minute. Katie, we do both clinical assessments, and what you see in white is pre-dosing, lameness, pain of various types, disability. What you see is one injection over spinal cord, and months later, all the pain measures continuously go down. And this is what we've seen across dogs, just as an example. So this is Katie. And listen for the owner's comments. Katie's a beautiful girl, but she's a mess. She's got so many problems. You can see the condition of her. Spondylosis is this merging of the vertebral columns. Disc degenerations, you're getting all the nerves that are getting squished and so on. Severe hip joint. If you've ever seen an x-ray, that's a nasty looking hip if you've ever seen an x-ray of a healthy hip. It's supposed to be nice and smooth and gorgeous. This is before treatment.
Yep.
Bunny hop.
See how she bunny hops?
Yep. That's because of hip pain. She can't independently move. And
Yes.
after treatment, the width.
I won't. I'm just getting her.
Now, independent motion, she no longer bunny hops. And it's been really insightful because I've videotaped.
Following me around more, which is way more normal. You know, she's with me all the time instead of
That's so good.
laying in a corner.
That's real as a parrot in the background.
Wants mom to go out.
Hey, go. I'm not sure about you. Come on.
Up the stairs and then back down the stairs.
Katie. What are we gonna do?
Yeah.
And what's really remarkable is, sorry, it was really loud at the beginning, might not have been able to hear. She was talking about, and she's followed me around the house now. I kind of describe it as the dogs get back their doghood. So if you know dogs, they play with toys, they follow their owners, they meet people at the front door if the doorbell rings, they hop in and out of cars, they take stairs. Chronic pain dogs don't do that. Katie was lying in the back room, didn't even come for strangers to the front door, didn't follow mom around, didn't play with her toys, didn't want to go outside. Took ramps when she couldn't take the stairs. After treatment, all of that came back. And this is the second example. This is one example from osteoarthritis because we're now doing an injection into an affected joint. And so again, this is an example of the veterinary assessment. Min-max pain, activity, quality of life, stairs, walking stuff. And what you see again is up is bad, down is good. And you see very long periods of time after single injections, a remarkable recovery. So this is one example of Louise. And again, listen to the owners. And if you can't hear the owner, let me know. I'll turn the volume up. I just don't want to push you out of the room with this. If you've ever seen an elbow, it's not supposed to look like a cauliflower. That's a really ugly, ugly elbow. Louise is part of our blinded study. So this is, while the owners did not know she was on placebo at the time.
Come on. Let's go. Come on.
Keep in
Go
mind,
outside.
the really bad one you saw was her left. So on the right of the screen.
Louise, come on. Let's go.
And if you ski at all, you notice she's traversing because it's a little easier to go up more slowly to get to mom to go outside. And this is a better formulation of the same treatment
Louise,
we
come
were
here.
talking about.
Come here. Come on.
Go on, Lise. Go get her.
Come on.
And up we go.
Good girl.
Up we go.
Much better, huh?
Yay!
Good girl.
And we also do this analysis on what you're about to see. This has thousands of force plates, but notice how nicely she's trotting along. It's just remarkable. Just remarkable. So the force plates that she's been running on, we can analyze what parts of the paw are being placed down, in what order, what the gait is like, how much force, how much weight, whether they're compensating because they have a painful joint and so on, and all these kinds of measures are improving. It's just really phenomenal. And do we have a few more minutes? It sounds like, it looks like we have. And this extends even beyond what I've been telling you about. So in other studies that are funded by the National Multiple Sclerosis Society, we've gotten into rodent multiple sclerosis models in hopes to also take it to people. These are very early days, but they are striking data that I wanted to share with you. And we got into this because about 60% of multiple sclerosis patients have central neuropathic pain. We got into it because of pain. But when we got into it, we fell into an observation we were not expecting, which was this therapeutic also calms and reverses paralysis. If you don't know this model, the videos are disturbing. You don't have to watch them. You can avert your eyes, but this is reality. It also tells you that this is really, really real. It's not just Linda's observations making up data in any way. So in this model, it's kind of like when you get a flu shot to prevent flu, you get an antigen, the flu bug piece, and you're creating antibodies against that to protect you. In this model, in rodents, they get part of a central nervous system myelin. That's what covers the nerves. That's what gets attacked in multiple sclerosis. And they create antibodies against their own central nervous system myelin. And as the antibodies begin to attack, in a rodent, it starts at the tip of the tail and more territory gets involved. So when you have a 0, that means they're perfectly normal. The bigger the number, the more real estate is actually being affected. And you get into this relapsing, remitting model, just as you see in humans. And to start the model, they don't get into an assignment to a group until they're showing symptomologies. This is what it really looks like. Rats are prehensile. They love to have good motor tone in their tail. They have complete paralysis of the tail. That is not a normal tail before they get assigned. And if they go into the group that got vehicle, they go into this relapsing remitting model where they now have a score of about 4 to 5 at the termination. The animals that got the therapy not only don't express as much paralysis, they go back to normal and they don't relapse. Now this video is difficult to watch. Turn your head if you don't want to see this, but this is reality. The animals that don't get the therapeutic are now paralyzed on the hind body, while the animals that had been paralyzed are now completely normal. This goes up to a score of 5 with an upper body before it terminates. So what have we said about this therapeutic? What we introduced this as is that you have an amplification of pain. We said that you have amplification all along pain pathways. These hot and bothered glial cells are releasing things that are activating pain. Here's the intrathecal therapeutic or into a joint if it's osteoarthritis. And as it creates this nice, cool blue interleukin-10, spreads across the spinal cord, calms down those hot and bothered glial cells, and pain goes away. And it's a beautiful day. So what do we conclude? The first conclusion may now seem pretty obvious and silly, but it's actually important, and that is immunology is important. And I say that because pain people have forever ignored immunology, and immunologists have forever ignored pain, and they're wedded. I mean, these are two fields that have to work together. You can think of these glial cells as volume controls. They do not have axons. They're not like neurons who can send messages from the spinal cord to the brain and so on. They're local gossips, if you will. They can turn up pain. If they're activated in drug reward areas, they can turn up drug abuse. If they get activated, they can amplify whatever's going on in their neighborhood. These glial cells do not care about normal everyday pain. So if you stub your toe or you cut yourself shaving or you smack your thumb with a hammer, your glial cells do not care. Your glial cells love you, but they do not care about normal everyday pain. Something has to trigger them to activate. And we know that they get activated physiologically as part of this ancient sickness response, which we didn't have time to really talk about. But if you think about the last time you had a really ugly flu and what you're going through, fever, sleep, changes in food and water intake, you didn't feel like partying till 4 a.m., going to a good restaurant be a complete waste if you had the flu. And in fact, what God discovered was these glial cells are creating all of that. And that's how we got into this originally back in the early 1990s, was glial cells amplified pain as part of the sickness response. Even the bed sheets hurt when you have the flu. Pathologically, when triggered by peripheral nerve injury, cancer, and so on, pharmacologically, by every single class of clinically relevant opiates. And in every case, these glial cells are becoming activated. They're linking now to tolerance, dependence, drug reward, abuse, and so on. And these pro-inflammatory cytokines are key. So we're hoping that targeting these glial cells and their glial cell products are going to be both a novel way of now treating chronic pain, but also making clinically relevant opiates work better for things like pain control. I'd like to thank a couple people in closing. I have an incredible team of people. Rob Landry is an amazing veterinarian that we're working with. Salute Therapeutics is a company I helped found, that's bringing it forward to the FDA. Kenna Rice and my NIDA, National Institute of Drug Abuse collaborators. Aaron Milligan started the interleukin-10 work. Peter Grace has taken the priming work. And where are you? Mark Hutchinson started the toll-like receptor 4 work in my lab at UC Boulder. Thank you very much for your attention.